Hey everyone, in this video we'll be discussing primary transcript processing, also known as pre-mRNA processing or simply RNA processing. We'll start this video off by talking about what RNA processing is. We'll then go on to talk about the three different changes that takes place to turn a pre-mRNA or primary transcript into a mRNA. And lastly, we're going to discuss some of the differences between protein coding transcript and different types of RNA. So let's get started. What is RNA processing? RNA processing are the modifications to primary transcript which ultimately lead to the mRNA. The primary transcript is the final product of transcription. So after transcription, the RNA product is known as the primary transcript or the pre-mRNA, which is then modified to make the mRNA. Now there are three main modifications in protein coding RNA that take place in eukaryotes. This is the removal of introns, the addition of the 5' cap, and the addition of the 3' polyadenylate tail. Now in prokaryotes, there's little to no modification of the primary transcript. This is because in prokaryotes, there is no nucleus. So as transcription is taking place, the primary transcript is being made, which can be translated directly. One of the first two changes that occur as a part of the primary transcript processing is the addition of the 5' cap. This is the addition of a 7-methylguanosine to the 5' end of the RNA chain. Now if you note here, this is unlike a regular phosphodiester bond, as the 5' carbon of the sugar of the 7-methylguanosine binds to the triphosphate of the 5' end nucleotide's 5' carbon. As you can see right here. So the 5' carbon of the 5' end of the nucleotide is attached to 3 phosphates, which bind to the 7-methylguanosine's 5' carbon sugar. Now what the 5' cap does is it stabilizes the primary transcript. It also stops the 5' end of the primary transcript from participating in bonds with other molecules. And the 5' cap is also important in the recognition of the mRNA by the ribosome. The next modification that takes place is the addition of the poly-A tail, also known as polyadenylation. This is the addition of around 250 adenosine-based ribonucleotides to the 3' end of the primary transcript. Now, much like the 5' cap, this poly-A tail allows for the stabilization of the primary transcript. However, it also plays a huge role in the movement of the transcript outside the nucleus into the cytoplasm where the ribosome is. So it allows for the transport of the primary transcript outside of the nucleus. The third and last modification that happens to primary transcript is the removal of introns. Introns are non-coding regions within the primary transcript that interrupt the coding regions, also known as the exons. So the exons are the coding regions within the primary transcript, and introns are the non-coding regions of the primary transcript that we must remove. The introns are removed through a process known as RNA splicing. This occurs through the action of a spliceosome, which is a bunch of RNA complexes bound together. Now the spliceosome binds to the intron and causes a site within the intron to attack the 5' splice site. This forms what we call a lariat. The lariat is just a looped intron. This also causes a free 5' exon. Now this 5' exon has a free 3' end that ends up attacking the 3' exon's 5' end. What this does is it releases the lariat as well as the spliceosomes and results in the exons binding together. Now this process repeats throughout the primary transcript where the introns are removed and the exons bind together. A good analogy to think of this is if the primary transcript was your plate of food, the introns would be non-edible portions within your food. So you must remove the introns, you must remove these non-edible parts of your food to be able to eat your food. Similarly, cells need to remove introns to be able to translate this message. When it comes to RNA splicing, an event can take place called alternate splicing or alternative splicing. This is when a single primary transcript can be spliced in different ways to yield different products. In this example, this primary transcript has exons E1, 2, 3, and 4. With alternative splicing, we can have either E1, 2, 4, or we can have E1, 2, and 3. Now each scenario yields two different proteins. This is very similar to remixing a film or remixing a song. The same song can be used and remixed in two different ways to yield two different products. In the same case, the same primary transcript can be used and spliced differently to yield two different proteins. Last but not least, I wanted to note that different primary transcripts could be modified differently, depending on the type of RNA. In the case described, we were assuming that the RNA is protein coding. We won't go over the different types of RNA modifications, however, we will touch upon each type of RNA and their functions. Ribosomal RNA, or rRNA, is found in all ribosomes that aid in translation. Its genes are found in the nucleolus. Transfer RNA, or tRNA, carries individual amino acids for translation. Small nuclear RNA, or snRNA, is found in eukaryotes and is involved in splicing, polyadenylation, and other processes in the nucleus. Lastly, microRNA, or miRNA, and small interfering RNA, siRNA, are regulatory RNA molecules that can destruct certain RNA transcripts. I really hope this video was helpful, and I hope you have a great rest of your day.